nucleus, genuclear nucleus. That's the place where the optic nerve is connecting to the optic tract. So this is a place called genuclear nucleus. And the tumor compresses the genuclear nucleus over here. And this patient has a symptom. His symptom is his left upper part of the eye field is blind. He cannot see his left upper part of the eye field. 左上方的视野缺损。The reason is this, because of the tumor compressing this part, this place, and causes the left upper quadranopsia. Okay, so this is very important because when you see a patient and he complains that he cannot see something in his left upper corner, you may think, oh, maybe he has problem in his visual cortex. Maybe his visual cortex has some problems. But actually, no. The problem is not the visual cortex. Visual cortex over here. Visual cortex is not affected at all. The problem is over here. The tumor compresses the lateral genicular nucleus and causes the symptom. So we can see, we start to see, using diffusion tratography, it can tell us the answer on why the patient has this symptom. And we can also study many interesting things in the brain. And this is the paper we published 2 years ago. And this is monkey brains. They are all monkey brains. And we did a very high resolution, high quality diffusion spectrum imaging in these monkey brains. And we start to see some organization patterns that we never saw before, which is actually those fibers, they form a very highly regulated grid pattern. There is perpendicular grid patterns. Every place in the brain. So at any place you can see they crisscross each other in a very highly organized way. So this is the finding we found. And now we start to see the same grid pattern in the human brain. And we still don't know the reason why the brain shows this highly regulated crisscrossing pattern, like the grid, like crystal. Okay. So here we have a National Geographic in 1987 cover, and at that time, medical imaging is like this. And this is this month's National Geographic, and this is the brain fibers we produced. So you can see the difference. It's been like 25 years, okay? And this is the 25 years ago, and this is today. When I was in 1987, I was a radiologist, first year radiologist. At that time, I still remember, the imaging is very primitive, very, very rough. But after 25 years, the imaging is very advanced. We can see many, many details, many, many interesting things using imaging techniques. I wonder, after 25 years from now, what the imaging will become. I wonder, yeah. And this all depends on you. So we can, actually because the time, I think I need to stop because of the time. So I want to show you what my recent work. Actually, I try to image the brain, and then I average hundreds of brains in a very sophisticated way. And then I determine the brain fibers actually, there are very nice organization of the brain fibers. And here, I'll show you the movie of the brain fibers. This is called association fibers. These fibers are all going anterior, front to back, front to back. Okay. The computer is a little bit slow. So this is the second system of the fibers, called commissural fibers. These fibers are going left and right, connecting two hemispheres of the brain. 电脑有点慢，所以到这边就卡住了，然后变得很难看。And this is the third system, projection fibers. These fibers are all going up and down from the brain to the body. Okay. And the reason why we put a lot of efforts by separating all these fiber tracts is actually we want to develop a automatic sampling of the tract integrity of the whole brain. So that's the reason why we do this. And this is the flow chart of how we can sample the brain white matter integrity using our fiber tracts. So we can get the data like this. So here we have an array of the data. So you can see, so we have, for instance, we have 74 tracts in the brain. And for each tract, we have 100 steps along the tract. And for each step, we can sample the FA value of the tract. FA represents the microstructural property of the tract. So we can have a 2D array for each subject. And if you have 100 subjects, you'll get a 3D array like this. So this is a very concise, standardized data of the brain fiber integrity. So if we have this kind of data, we can easily compare the patients and the normal subjects and see how different they are, where they are different, what kind of tracts are different. Yeah, so we can do all these kind of studies. And actually, we already have some results by comparing patients with schizophrenia, 精神分裂症, or patients with autism, 自闭症. And we found that there are many tracts changes in the patients. And these changes are actually associated with their symptoms. And we start to find these kind of interesting findings. So that means this tract study actually is to study the connection of the brain. And many mental disorders like schizophrenia or autism or ADHD are actually the disorders from the connection. 线路不大对劲. And we can now detect what's going on of their connection. Yeah. So this is called connectome study. this connectome is a term just recently popular in recent 5 years. And connectome means connection. And connection means you need to study the connection in the brain. And this will tell you many information about the brain, about the disease, or about the genes. Many genes will also affect the connections. So this is a very interesting area to work. So conclusion. I hope I can explain to you the diffusion MRI technique and let you know that this technique is very interesting. It can detect cancer tissue and also reveals ingenious design of the fiber architecture of the heart. And also we can use it to analyze the connectivity of the wired brain. So diffusion MRI is an interesting technique to work. And it's still evolving. It is still evolving. And actually many, I know many scientists, they are physicists background, and they are now working on diffusion MRI. Yeah, okay, thank you very much.
Well, it is really very impressive that we can now map out the tissue in our brain and in our heart in such great details. Okay, so any questions?
Is there did you find any major difference in the normal people's left and right hand side brains?
Okay. Yeah, actually, we did a study, but not published yet. But I think, yeah, because left-handers and right-handers, so left-handedness and right-handedness, actually, the fibers in the brain they are different. They are different. For instance, in the right-handers, maybe around 95% of the right-handers, their language network is in the left side. But in the left-handers, maybe only 75 to 80% of the left-handers, their language network is in the left side. That means that there is some difference between the handiness and the language network. And we found that for the language network, we can analyze the fiber tracts in the language network, like archaeophysics. It's a special terminology, but there are many fibers involved in language network. And when we study those fibers, we can see the difference between left-handers and right-handers. Yeah. So I can promise you there is difference, although we haven't got a complete data yet, because left-handers are not many in Taiwan.
Any other question?
Very good question. Actually, we are also interested in the difference between men and women. How the brains are different. And we also found that they are indeed different. In a tract called fornix. Fornix is the tract involved in emotional processing and memory. And we found that women, fornix tract is stronger than men's. So that means women shows more emotional and also maybe have better memory than men.
I have a question. The fiber you are talking about, What's its relation with what we know as neurons or things like that in the brain? Is there any connection at all?
Okay. Yeah, this is also a very good question. Actually, from the bottom up, if we look at the microscopic sizes, we will see neurons, and then we will also see the axons. Axons are the extension of the neuron, and each axon will connect to many neurons, and then they do the neurotransmission, signal transmission. So axon actually is very thin. It's like 2 microns in diameter. Yeah, and the diffusion MRI, we see the fiber direction. This direction actually is the average of a bundle of the axons in that location. Right. Yeah.
Yes. So what's the resolution, spatial resolution you have for the brain, and what's the resolution you prefer to have in the future, and what's the limit there?
Yeah, a very good question. This is a question from experts. Right now, the spatial resolution of our diffusion spectrum imaging is 2.5 millimeter cube. 2.5 millimeter cube. I prefer the resolution is to be 1.5 millimeters. I think at 1.5 millimeter cube, we will see more smaller fibers. Yeah. And what's the limit? Yeah, there's a limit. If I can have some magic power, of course I want to have 1 millimeter cube. That's perfect, right? But I cannot do that because once you have smaller and smaller voxel size, that means the element of the image, you have less and less signal, right? And this will kill you, because no signal you will see nothing. Okay, so we need to have sufficient signal, and in order to have sufficient signal, we need to have a larger voxel, and so we cannot have unlimited small voxels, as small as we want. So I think 1.5 millimeter is what we can achieve in our current technology. Right.
Any more questions? Yes?
Okay. Is it possible to do a whole body diffusion MRI if given long enough time?
Yes, this is a very interesting question. And actually, it is feasible. It is possible. For instance, there is already a technique. It's called whole body diffusion weighted image. As I show you in the prostate cancer, right? In the cancer tissue, the diffusion coefficient will be decreased, ADC decreased, right? And then you can apply this technique to whole body, to image the diffusion weighted image of the whole body. And then you can screen for cancer. Do this to screen for cancer. So this is already a product now. Yeah, it's initially developed by a Japanese. He works on the MRI, and he thought of this idea, and he actually developed a whole body diffusion MRI, a diffusion weighted image.
So how long do you take this kind of?
Not too long. Actually, the sequence only takes about maybe 10 minutes or 20 minutes.
Now, what's the resolution? How big is the cancer cell that it
Right.
can detect?
I think the resolution is not very fine, not very fine. But all you can do is to detect like lymph nodes. Yeah, lymph nodes. The lymph node of about 1 to 2 centimeters. I think it can detect whether there is cancer cells or not.
Okay, if there's no more question, let's thank Professor Tseng for wonderful talk again. Thank you.